Good morning, everyone, and welcome to today's MPG Primer. And today it is my pleasure to introduce or reintroduce Sarah Morton. Sarah Morton has been a pivotal member of the MPG community, having served as the co-chair of this primer series for several years, and has some interesting outlooks about what she'll be doing next with MPG. So hold tight for that. So just a little bit about Sarah Morton. She is a neonatologist at Boston Children's Hospital and an assistant professor of pediatrics at Harvard Medical School. Her research group is focused on genetic contributions to congenital heart disease and associated health outcomes. She is also the director of the NICU Biobank at Boston Children's Hospital and is glad to talk to you today about biobanking big and small. Thank you so much, Sarah.
Thank you so much. Thank you all for being here today despite the rain and slush. And so I'm really glad to share with you a little bit of what I know about biobanks, but would love this to be interactive. So if you have questions or thoughts as we go along, I'd be really glad to engage. But I think the perspective I was thinking of is, if primer series is for people who are, maybe earlier on in designing projects and thinking about our scientific community, what is it that you might want to know if you wanted to set up a biobank? What is it you might want to know if you're accessing a biobank about how the data are obtained and the decisions that are made in the setting up? And then I'll give you the example of our local biobank, which will have the definite theme of everything in pediatrics is smaller, but I also think that sometimes our effect sizes are larger. So there's something to be learned from kind of every type of cohort. And so through the talk today, we'll talk a little bit about the general principles of biobanking, and that's where the why, what, who comes in. I'll talk about some of the exemplar ones that I'm sure you're all familiar with, but what I'll be sharing with you a little bit is like, what does the protocol look like for all of us? What did they decide to do with samples for UK Biobank so that you can see what are the trade-offs between scale, cost, and kind of preserving questions that can be answered in the future that we can't even anticipate now. And then I'll tell you about our local one over at the NICU. So what's a biobank? What's a biorepository? We kind of all smush the words together. Is there a significance to the different ones? And I would say a biorepository is a larger umbrella term that is when there is a banking or kind of a storage of tissues or samples that come from any kind of living organism. And so often when people talk about a biorepository, it might include bacterial cell lines or tissue culture lines from non-human animals. And so a biorepository is a more general term of which a biobank tends to refer to biorepositories that only include human samples. And so the other thing that makes a biobank is not just that you've stored it. So having a freezer is not necessarily a biobank, but that the specimens were collected for the purpose of advancing medical and health knowledge. And that's important because you can't just have a sample. You need to know who it came from, what were the situations in which it was collected, perhaps even what has happened since that person participated, what are those longer term outcomes. And so what makes a biobank is not just the sample, but the metadata that come with it. And sometimes you might spend as much effort and as much of your budget on getting those associated metadata as you do on the actual processing and storage of the samples. The other thing that makes it different is that there are kind of standard operating procedures and a set of governing principles that you should adhere to when having a biobank. And so there needs to be a standard operating procedure for the acquisition of the samples, for the processing and labeling of the samples, and for storage and distribution. And so if you are either trying to access a biobank or setting one up yourself, knowing how to enter each of those different phases and how to understand how a sample you might be using was processed is one of those things that you can really dig into in the metadata. My favorite thing about a biobank is always its data dictionary. Because if you know what they know, then you can also start to understand a little bit about what might be missing that you would need to infer or acknowledge as a limitation in your own study. Biobanks are so exciting right now, right? They're growing, there's national ones, there's international ones. Every hospital has one. They're adding metadata, they're adding EMR. You can do natural language processing, right, on medical records that are linked to biobanks. And so it really is an area where there's been a tremendous amount of growth, both in terms of the number of biobanks and the scope of their purposes.
May I
And
ask a question about
yeah.
that? What do you think are some of the reasons of why this is sort of booming right now?
I like to think it's because there's a lot that as we advance genetics, some of our animal models and cell culture models, which have given us so many insights into the fundamental biology underlying health and disease, that we've come to realize that a lot of the fruitful advances recently have been very specific to the human genome and very specific to human physiology. And so I think that some of the boom is the focus on moving towards understanding our fundamental biology, but also moving towards having new therapeutics or new diagnostics in the near term. And so I think biobanks are a nice opportunity to bridge our basic science and our cellular and animal models as a nice demonstration of applicability before you go to like a phase 1 trial or something like that, thinking about developing therapeutics. So I think of them as having a nice role in making you can kind of take all of your science and make sure that it holds true in a human population. Yeah, and so this is not a tough crowd to say population genetics is important, right? But like having these broad, diverse, representative cohorts really lets us interpret variants. Like when I think about, we do genetic testing on patients in the NICU regularly. At least once a week, someone gets a genome sent. And our ability to interpret those variants now, because your allele frequency can go down to 10 to the -5 or 10 to the -6 in the general population, right? Those kind of data come from our, in large part, for efforts of our biobanks. And also, just having many biobanks, as we'll talk about, will be able to share with you cells and perhaps even the cells that can be made into iPS cells. And so that does let you look at molecular mechanisms among people who you might know their genotype as well as their kind of medical history. And then, again, switching and thinking about interventions, it's a great place to look at questions like pharmacogenomics, where we've come in the past 5 years to understand so much more about how our generalizations from a limited population are not really representative to our entire patient community. I just thought this was really cool. That's a picture of like the inside of the UK Biobank. It's just from their website, but really thinking about how sophisticated many of these operations are with almost no one touching a sample once it's been collected, right? And having the automation both to reduce our amount of potential contamination and differences in handling, but also really to allow a scalability that's not possible without automation. I came up with my title before I found this paper, but I found it very interesting that we had the same kind of way of breaking down how to understand a biobank, right? Which is, if you're really thinking about using one or thinking about starting your own, you need to figure out what your scope is. And to me, these same sets of the 5 W's plus how at the end is a great way to define which biobank might be right for you or how you might start your own if you needed to. And so we'll go through these more in detail, but really why is obviously the first one. So either why would you access a biobank sample or why would you start your own, usually has to do with, as Nicole asked, like what is that key question that's where there's a gap in your science, where either having a biospecimen, having metadata or aggregated or single information from a population would answer your research question. The what is one of the things that changes the most. We have a biobank over at Children's right now, and they're just going through the process of pretty much only keeping white blood cell pellets. So there had been a lot of serum or plasma that was stored for a long time, and it turned out those just hadn't been accessed by researchers, right? People were interested far more in the DNA. And so as the scale of the samples became large enough to require expansion or selection of samples, they chose selection of samples, right? So the what definitely changes over time. And a lot of the what is electronic now, right? It's your wearable device data, it's questionnaires filled out by participants, things like that. The when part, I think, is uniquely interesting in terms of thinking. We used to think of a lot of our research participation as a single event. You fill out one questionnaire and give one sample, and that was your participation in the research study. And now it's become much more longitudinal, and people are more willing to share a lot of their personal information in an ongoing way than they were in the past. And I think, though there will be differences in who contributes data and who has time to contribute data that we always need to be cognizant of, it definitely has expanded the when of interaction, especially with passive drawing of data from the electronic medical record, right? So that requires you to just return to care from the place you enrolled. But that's another way to get kind of longitudinal data from individuals. We'll talk about the where, especially when we talk about all of us, but that's been another place where, you know, it used to be hospital-based or institution-based, but there's been a lot more biobanks with a community focus in terms of outreach and enrollment, where that part has changed. And then I think the who, from a pediatrician, one of my biggest perspectives on this is everyone I enroll because their parents consent, I would need to re-enroll once they reach age of majority. And so from me, most of the who's, people can always opt out. But one of the things to think about when approaching pediatric populations is there's actually an active need to opt in once you reach age of majority. We don't, just because their parents said yes when they were a day old, doesn't mean that they, as an adult, will say yes themselves. And so that's one of the things I think about most with the who, is just making sure that we, you know, not just passively accept people's choices to opt out, but there is actually a need to proactively confirm consent. So if you think about why to start a biobank, why did I start the NICU biobank? Why did people start UK biobank? There's very different motivations, right? Depending on kind of your scale and your catchment. Sometimes it might be started for a specific project. For example, many times there might be patient registries that are associated with a clinic or an organization that represents people who have a particular diagnosis. And so those registries are now often expanding to also include a biobank. Those are often optional, and that the primary project is still the registry and collecting non-biospecimens. But sometimes biobanks are an add-on to a project that's been going on for sometimes decades, but now collecting biospecimens in addition. And especially with the advances in ability to do nice genomic sequencing from saliva samples, this doesn't even require anyone to see a needle or show up in your state. And so that can be a way that people do expand biospecimens. We're doing this right now for a study of adults with congenital heart disease who are now in their 30s and had their operations generally in their first week of life. And it's been amazing how they participated in this research study over decades. And one of the ways that we've gotten biospecimens for that study is literally we just send them a spit kit and they send it back. And then all the sequencing happens here. And so it's a nice way to be able to expand our data types on studies where we might actually already have that longitudinal information, but we've never been able to consider the genetics in the questions. Sometimes there might be a planned analysis. I'll show you an example of this later. I tried to make my our biobank such that, you know, I last night when I was working in the NICU cared for 2 infants with trisomy 18, right? And that's not everyone's every day. So I am very privileged that I get to meet a lot of families and infants who are affected by diagnoses that are rare in the general population. And to me, it was always a tragedy that a fellow might show up 5 years later and want to study trisomy 18, but we would have no samples for them, right? There would be no place for them to start from, even though we had, you know, some of the most ability to start collecting those samples. And so we've made our IRB such that a fellow can come with a question. We will do all the consenting and sample collection, and they do a secondary analysis of available research consent. And so that process becomes much more easy for them. And then we also have just, you know, the accumulated knowledge of the years that it takes to get a reasonable cohort. So that's an example of a time where there might be a planned analysis, but we're just kind of adding on the specific data types that we need. Then there's the more general projects. So these are the ones we're familiar with, the UK Biobank, the Japan Biobank, all of us. And so there's often broad participant recruitment. This is where the metadata might be a little more variable. It might fit your study, but might be missing some pieces, right? Like in the NICU, there's all these things we collect about what was maternal, what was the health of the pregnant person during pregnancy, what were the incidents that happened right around delivery, birth weight. It's amazing how hard it can be to find someone's birth weight. Even in our hospital, sometimes if they transfer to us 2 months later, it might never enter our medical record. And so there's a lot of more details that we catch in our targeted metadata, which might not be present in the general projects. And then there's a lot of times where people come up with cool new techniques like episignatures of different congenital anomalies. And all of a sudden we already have this biobank. And so we didn't know that that's what we were going to be doing with our samples. But now that as those technologies have progressed and the research questions come to encompass our population, we're able to then set up the appropriate agreements to be able to transfer those samples. So who is in the biobank obviously determines your ability to learn from the samples in the biobank. And so one thing is always read the fine print. And so if you read what the inclusion and exclusion criteria are for a biobank you're thinking of accessing, that can help you know at the start what the caveats will be to your analysis. Again, I mentioned this before, but just anyone who's enrolled under the age of 18 tends to need active reconsent now. That's a change in the past 3 years or so. It used to be that people didn't really require that reconsent, but pretty much everyone now is having their IRB protocols revised to include that reconsent. And then just a couple of ways that we do in our biobank to be representative. Is we have all of our research approaches be regardless of preferred language. Our hospital actually does not have any charge for use of interpreter services for research. So there should be no barrier to enrolling anyone or approaching anyone for research who's eligible, and then pursuing a consent if they are interested in hearing more. And I actually think that's a really important part because many protocols that still come to our IRB will say English only or English Spanish only because it's a pilot study. But there's actually a really nice thing called a short form consent. And what that is that there's a much shorter form of the consent, which is translated by the interpreter. And then the larger consent is verbally translated in the moment. And so there are ways to be able to not have to have your documents in every single language, but still be able to provide a robust consenting process that doesn't limit participation based on preferred language. Things like, so this is again, maybe a little more NICU focused, but not everyone's going to be available to stay an extra half hour after their clinic visit. Not everyone's going to be available 9 to 5. And so having things like off hours consenting, having remote consenting, which has been, were there any silver linings to everything becoming remote for a while? Being able to have some online consents where people can read at their leisure. There's no one in the room. They can take their time to ask their questions, but be able to be asynchronous in some of that can be another way to be more inclusive. And then also I think I know why I would participate in a research study. I dragged my kids around to be people, they had so many of those like motion balls attached to them for some study, right? Like many of us participate in research for all kinds of different reasons. And what actually matters is not why I participate, but why other people who have chosen to participate in that biobank or that research study. And so I think as much as possible, having the voices of participants represented in your brochures, in your flyers, even, like on the screens that are around every place where someone might wait for their consent, having the voices of people who really participated and why they did can be one of the most compelling things. And so I think that's something that can also help you have a more representative biobank. And then just thinking about, again, location, if you only recruit in our NICU, you're not going to get the average neonate, right? And so we're really good at answering some questions and terrible at others. So just making sure that if you're going to access a biobank, understanding how it was that people were approached, because it's, again, when it's location specific, people usually either were there or not there for some underlying reasons. So what is banked? This it talks about all the different things there. The image is talking about one that was an oncology example. And so I liked it because it focused on the participant pathway. So that's their participants starting at the bottom left. And so people often provide a lot of things. They'll provide biosamples. Some of them are passively collected, like our biobank at Children's Hospital. We're actually really good at using not very much blood for testing. It's not quite Theranos level, but we do things on very small volumes. But they even save the rest of that very small volume because they know that some of my patients are 500 grams. You can't take a lot of blood from them. And so with that, they'll actually save whatever is left after the test was run in case I want to add on another test. Like, oh, I needed the liver function, but now I'd really also like to add on, my albumin or my electrolytes or something like that. And so they already had this culture of saving the remnants. And so all you had to do was say, well, after you've reached that remnant part for retesting, how about that just goes to our biobank? And so there are ways to participate that don't require extra needle exposures or extra biospecimen collections. Similarly, sometimes operative tissues are the most valuable, right? Because you might be studying a condition that is not easily accessible or directly studied from blood, CSF, or urine, something else that comes out of us more easily. And so having that pipeline to be able to access surgically excised tissues is also a high yield place. And so again, those biosamples might be population-based, in which case you're grabbing whatever's left over or generally taking blood, or they might be very disease-oriented, in which case you really want that tumor tissue that was removed, right? Or you really want the cerebral spinal fluid, because what you're looking for is really a central nervous system process. The demographic data, which is now more and more passively collected from electronic medical records, but obviously can be from a survey also. Clinical data is really interesting. I spend a lot of my time with ICD codes. ICD codes were meant for me to be able to bill, for me to say to the insurer, this patient's really sick or this patient's not that sick, right? And that's how we communicate with payers to justify the hospitalization or procedures or whatever else there is. They were not intended for research. And the things I say match the current condition. Someone might have respiratory failure. They need a ventilator, but it's because they just had surgery and they had anesthesia and need to recover. It's not because they primarily have failure of their lungs. And so there's a lot of things like that where what you're actually getting in ICD codes is secondary and not primary. And those codes don't always discern those things. So my biggest lesson or my biggest message about clinical data, it's like buyer beware for your ICD codes. And we've looked at things like defining ADHD or autism spectrum disorders using ICD codes across 9 centers of our congenital heart disease study. And the diagnostic rate ranges from 1% to 60%. And I don't think it's because the people who are receiving care are different, right? There's differences in how the codes translate to words at each hospital. There's differences in how many codes people click. And so that's one of the things about clinical data is I was always just kind of, as much as you can do a little bit of ground truth checking, even if it's at a single center or a couple of centers, or use something like natural language processing as an orthogonal measure, that's one of the important things to think about for clinical data. There's often directed study evaluations. They might be, directed neurodevelopmental testing. We do a lot, right? So we'll do these tests called the Bailey test, which is how you know someone's fine motor versus gross motor versus cognitive performance. So there can be directed study tests, and those are often the ones that are most high quality because they'll many times be standardized, right? Where everyone who's administering the test has gone through the same training. There's some cross-checking where the test might even be administered by 2 different people for some period of time to make sure that there's a lot of cohesion. So many times it's those directed study evaluations that are the most harmonious to use between centers or over time. And then the questionnaires, which are just lovely because many times that's the only way you get the participants' experience of life. Right? How do you know someone's quality of life? How do you know someone's, job satisfaction or, social satisfaction? It's not going to be from directed testing, right? And many times those outcomes aren't necessarily what studies are designed to report, but can be a really significant thing, especially when, again, thinking about pediatrics. It might not be whether your IQ is 82 or 86 that matters. It might matter like, what's your degree of completion of your education, what's your degree of autonomy as you've become a teenager, things like that. So that's where those questionnaires sometimes actually do have better information than, or different and more applicable information than some of the questionnaires or some of the directed study evaluations. And so if you do get your biosample, there are really expensive, really fancy ways to handle biosamples, and there's really simple ways. And they just all have trade-offs, and right, it depends somewhat on your resources and your scale. So when you're running a large biobank, people have as many automated steps as possible because that minimizes any contamination. Spent a long time looking at 16S sequences of samples we had collected in Uganda, transferred to Pennsylvania, sequenced, transferred again to New York, proteomics, and there was just so much decontamination you had to do from every step that was handled. And we saw as many batch effects from each step of the handling as sometimes we would see biologic effects, right? So really automated handling and minimized repackaging is really beneficial. The most common things that are stored are plasma, serum, and DNA. Some people, like the Children's Biobank now, are moving towards what's called the buffy coat. It's just that it's white and fluffy looking, but it's basically the white blood cells. And that's because if you get blood, the nucleated cells are the white blood cells, right? And so that's where your DNA is going to be. You don't need all those red cells. They don't have that information. And so that's why people are often now moving to just saving the white blood cells. Sometimes the primary bone marrow cells are also saved because those, if stored appropriately in liquid nitrogen or colder, can be turned into iPS cells in the future. So people like those as like a way of being able to access that particular genotype again. Other things can be urine, which is great because it comes for free, cerebral spinal fluid, which can often be obtained in a slightly larger volume during a routine procedure without any injury, as long as it's a small volume. And that's one of those ones that we try not to miss when it's available, because it does let you know very specific things about, brain metabolism or infections, things like that. And then the big thing, if you really do have a biobank and are meeting all the criteria, is you have to have very careful protocols about all the backups. And so you can't just have a freezer and slap a sign on it that says biobank. You actually, as I'm sure you've all seen, you have freezers with monitors on them that alert someone immediately if the temperature has changed. You also have to have a plan for like, if your freezer goes down, where do your samples go immediately, and things like that. And so again, this can be anywhere. It doesn't have to be a fancy biobank. It can be in a single lab. All of our samples that don't go to the main biobank just go to a shelf in my freezer. Like it's fine. They can go there. They just have to have the right protections and security. Sometimes it's a collaborative biobank within an institution, of which we know of many, right? There's the MGB biobank, which is lovely. Children's has ours. There are these national and international ones. We've accessed a lot of these to be able to get cardiac tissue, right, for transcriptomics, because not everyone saves cardiac tissue. And so that's where some of these big international and national banks can be really useful for rare samples that many researchers can use. And then just if you are a biobank that's storing any intact human cells, you need to be in a facility with a Human Tissue Act license. And so that's why when you're filling out your protocols for these things, there are a couple more requirements, but it's actually not that onerous to meet the requirements there. The major things are you have to make sure you can link your sample in your consent, right? You can't keep anything that you can't prove was consented. You have to show that there's that tracking of samples and fail-safe measures, and then security in the building as well as the local freezer. The metadata is one of the most variable things. I just last night was assigning Mondo terms to everyone in our NICU biobank, of which Mondo doesn't cover a lot of like neonatal disorders I was learning. So there's there's the ICD codes we talked about, which are billing codes, which are meant to talk to payers. And so some people have tried to develop other codes, which are more designed for research and better capture someone's actual, molecular mechanism of disease, if you think about grouping in that way. And so there's often raw clinical data that goes in, as well as these other kind of like metadata labels that kind of accumulate the information from the chart. There's some really nice ways to use REDCap to just capture data directly from the EMR, which is always the most accurate. It's kind of like the automated handling of metadata. So instead of someone typing something in and missing a decimal point, you can actually just have it directly pulled. They still might have missed the decimal point when they first entered it, but it at least is one step of less contamination. There's many de-identified de-identified portals where you can actually do things on the EMR directly. The data might be raw, it might be curated, and knowing the level to which it was curated can be helpful. And especially if there's a phenotype that's already been studied in that biobank, reach out to that group and get their phenotype labels, right? Because you hate to have a different result from the same cohort only because you had a slightly different definition. And so even if what you're going to do is purposefully different, understanding the difference in your kind of labeling of a similar diagnosis can be helpful to contextualize why your results might differ from someone else's. And then again, this is where sometimes disease-oriented biobanks will have very different clinical data available, and that can be the benefit of maybe going back and forth between a more specific biobank and a more general one in terms of maybe the power you get from the increased metadata and granularity versus the power you get from a large population. And so I think the two can work together nicely. And then you always want to know about the sample. When was it collected? Like we're collecting placenta right now. If you collect it more than half an hour after delivery, it's like the pancreas, it just degrades. And so you want to know when that sample was collected. Some things it doesn't matter as much. Cardiac tissue we've worked with up to 72 hours after it became available that it was collected, and that's been just fine. But really knowing for your sample what that time was, and then making sure that that time was not a factor in what you're measuring later to know for yourself that there wasn't a quality difference. Any processing, right? RNA later was all the rage and now it's terrible. And so knowing if it was stored in different kinds of media, even if that was later removed, can really impact your ability to interpret any kind of sequencing or other processing that you do after. The amount, many times there's a standard allocation. So after the sample arrives, it might be split into 5 aliquots. And so just knowing like how many aliquots remain for that sample can be important for making your plans, because often there's like an approval that's needed if you're taking that very last aliquot. And so that's often an important metadata. And then the time between collection and freezing. Then there's all the study-derived data. There's a lot of measurements now. And as I mentioned, this is an area that's just exploding in terms of people giving all their digital device data. The sequencing, and many times it's actually a requirement of accessing biobank specimens, is that you return the raw and processed data back to the biobank so that it can be generally available. Sometimes there's holds where you get exclusive access for 1 year or 2 years after a study period, but then it comes back to the whole community. And that's been a common way for biobanks to be able to grow their resources while still allowing the researchers who are funding the sequencing to have time to complete their analyses. And then all the questionnaires that we had talked about. So these are just some of the kinds of biobanks that I think about, and we'll cover very briefly. One of the oldest ones are actually these human umbilical cord blood biobanks, right? So if you go to deliver your baby, people will ask, oh, are you planning to bank the cord blood? And so literally what happens is, right, there's this long umbilical cord that connects the infant to the placenta, and they can just put a clamp on each side and it goes in a freezer container and off to the biobank. And so the public ones are then accessed by individuals who might need some sort of stem cell therapy in the future. And since you are usually unable to donate to yourself, it's these public banks that allow for appropriate cells to be found for individuals in need. They're also available for research. And so this was one of the first examples of like large scale biobanking. There's 28 public ones in the US. And so that's one. And so this was just showing the number of people per year who had who received a treatment using the publicly banked cord blood from deliveries that people had donated. So it's, in 2011, it was 1,000 people that year. And just seeing that it was benefiting a good number of people over 16 as well. That's the green part of those bars. So it's, benefiting not only children, but adults too, to have done this banking. That's all anonymous and public. This is just from the All of Us. They have very lovely documentation. Again, if you're going to use a biobank, if you just go onto their websites, they have all their protocol documents there. So you can see like what's actually the standard operating procedure, how are people approached, what's the sample handling. And so one of the unique things about All of Us was just this idea that there was not only recruitment at hospitals, but also within the community. And that was part of how they made sure that their participants were as representative as possible. And then the other thing was that this could be done, a lot of it can be done online. So whether you were at home or at one of these centers, you could go online, create an account, give your informed consent, contribute all the data. Again, you didn't have to sit in someone else's room. You could sit at home and contribute all this data. You would then go into your appointment, which I think I'll show you on the next slide. And you can also view your own data and results from that too. This is an example from the UK Biobank. This was a little older. This was from 2007. I couldn't find their more up-to-date protocols. But they specifically said that their aim in everything they did with biosamples was to maximize scientific return within budgetary limits. Which I think is kind of what all of us try to do all the time. But they took 45 mLs of blood from their participants, and then they separately processed plasma, serum, white blood cells, and red blood cells so that those 4 sample types were available. And so they also took 9 mLs of urine. In their protocol, they talk about why they considered a lot of other things, right? So, and this here, which I liked, was some of their reasons why they considered some sample types, but then excluded them. And so, hair, people think a lot about longer term environmental exposures with hair, but they just didn't know how to get it to, like it just seemed like the processing was higher than the benefits at that time. Saliva, they weren't choosing then. There are some salivary biomarkers. I think now we feel differently about that, knowing how much better our sequencing has gotten. And so, it's also nice that these protocols will often have some of that why. So even if you're thinking about starting your own, you can see how other institutions made their decisions. And then this was that All of Us study visit that I was telling you about. And so there was all of that participant provided information, which again, that part could be at home. And so because we often think about like, what's the burden of participation? And definitely the time spent at a visit is a high burden. We're often in places where you have to find parking. Someone's had to be with the rest of your family members. There's many things that come into an in-person visit. So reducing that time can really increase the ability of someone who's interested to participate. And so really what they did in person was the physical measurement. You have to sit for 5 minutes. It's like the white coat hypertension phenomenon, as well as if you were rushing there, right? So there was the 5 minutes of waiting so that you could get real baseline vital sign measurements, right? Because if your heart rate is high, but it's because you just got that very last parking spot, but it was 7 blocks away, it's different than if you have a baseline risk of tachycardia. And so then they would do their measurements and their biospecimen collections. And they very specifically were making these visits as short as possible in terms of the interface time. They also had a special pediatric protocol. And so this is, again, where you think about having a special subset where they were actually collecting different types of metadata. And so knowing, I think we all know that there's lifelong impacts of early health exposures, but sometimes it can be more useful to have those data if your overall participation also started early in life, so that you can track the impact of multiple of those different exposures. And so they had very specific questions about environment, about early life adversity that they collected as well. And so that was like a special cohort with a larger metadata set. And so what's our NICU biobank? It's a subset of our overall hospital biobank, which actually means that we end up having like 3 different versions of our consent, as I'll show you in a minute, because we try to kind of optimize someone's participation to maximize their potential benefit from being involved. Similarly, to kind of lower the bar of participation to meet where people want to be, we have many different tiered levels of consent. So it can just be the excess samples. So no extra needle pokes, not even a saliva swab, just whatever's in the lab and left over will be saved. We can do a separate blood draw. So you can have when there's going to be a blood draw anyways, we can take 1 extra tube and freeze that. Sometimes parents give us samples. We do a lot of genome sequencing for people who participate in our study. And so we'll often get parents' saliva is what we tend to do now, but they can also give a directed blood sample if they want. And then all of those things are with or without genetic studies. And so it's really that people can choose what's right for them. And it might change over time. Sometimes people are not initially interested in genetic testing, but then if they're still with us 1 or 2 months later, their desire for that type of information may have switched. So we do kind of check in with families as they remain there. One sample is stored for free by our biobank with the caveat that I have to share it with anyone who wants it, which is fine because that's why I got them. And any additional samples come to our lab. So first, yeah.
I have a question about your previous slide where you said that like over time they might change the permission. Do you also have it an option where they might actually take back permission? And then how does that like actually happen if people have already started like working with those samples or analyzing it? Like having to backtrack?
You have to kind of mark the moment that they withdrew consent. And then you do have to remove all of their data we remove it from the metadata. We discard their samples appropriately for biohazard. And then we remove all of their data from anything. You can't remove what's already been published, right? But you would, if there's anything that is not yet published, you would remove their sample from analysis. It's one of the reasons to have a really good consent, right? Like you want people to feel comfortable with what they have agreed to. And it's another reason to have a really good de-identification, right? We recently did a paper where we looked at genetic causes of hypotonia in infants across 5 continents, right? Sounds so fancy. But it really meant that we couldn't report anyone's variant because though it sounds like it's across a big population, it was 100 people. And those variants don't exist in NOMAD. They don't exist anywhere, right, that we know of, except for those infants sometimes. And so it's incredibly identifiable. And so part of it is also thinking like what you report. And though it would be useful to have the variants reported for scientific progress, and that's my inclination is to want to do that, we did not in that particular case because it was such a small number of individuals. So it is that balance, and I think always making your best decision. If you were a participant, knowing the range of participant opinions, how do you kind of best carry out what anyone consented to through the entire scope of the project? Yeah, so we were luckily already at an institution that was accredited, so we didn't have to do that part. We did get IRB approval, and then every time you change something, even if you're changing your brochure, they might ask you to change other things too, so that your entire protocol is up to date with current requirements. Because as you know, what's considered appropriate with genetic data is constantly changing. And so that's one of the things to know is that you'll be making lots of edits, but it's all so that you do the right thing. And sometimes it's just lots of back and forth. The other thing that goes, so there's a study protocol there, right? Which is like why we're doing it, inclusion, exclusion criteria, kind of what we're going to do as the study team. And then there's the informed consent, which is the version of that is captured, but for the participants to read. You need to represent what you're going to do. You need to represent what your aims are. You need to represent what the potential benefits and downsides would be. And then actually everything up there, everything goes in the IRB like a brochure. If you change who's on your brochure, you need to put a new version up there. If you're going to post on social media to try to contact participants, you need to have that on your IRB, and you need to have your posting on the IRB also. And so there's a lot of things. Some of the things that you might feel like are just automatic after you've gotten your approval are actually still looked at by the IRB. And then there's the things that don't necessarily go there, but it's the how to do things right. So it's the standard operating procedure that says, you know, Hannah, my lovely RA who does all of these consents, like it's how Hannah does it. And when Hannah goes to medical school, she will pass it on to the next person to make sure that they do the approaches in the same way, that they consult, you know, contact interpreter services in the same way, just so that it's consistent year to year. And if we make changes, we try to be very purposeful about it, because small changes in how you do things like that might impact who's recruited and whatnot. And so why did we do it in particular? And I'll be brief. is, like I had mentioned, I just think that there's a lot we can learn from our patient population. It's for both people inside our group, people in the hospital, and then external collaborations where it might be hard to get together, more than 2 or 3 people with a particular diagnosis at your hospital. But if we all work together, we can actually get a large group. A lot of it with all of these biobanks is to prepare for research, right? We have these transient contact opportunities. And so we wanted to start that contact. Many times funders will be glad to provide your sequencing if you already have the samples, having been burned for years by people who said, oh, it's great, look at my recruitment rate so far, I will show up with 5 million samples. And then those samples never appearing, right? So people often want everything to be in hand when you're making your proposal. And so this is one way of enabling that. There's these very rare events. And again, I think especially for trainees, being able to kind of maximize their impact in the time that they have is good. And then there's just questions we haven't anticipated yet. I get new questions every time I go to the hospital. And so being able to kind of have those things for the people who not just have the question, but are then going to act on it is good. And then as mentioned, sometimes it's just complementing ongoing research. So, the Pediatric Heart Network, which has always been a very clinical study oriented collaborative by the NIH for congenital heart disease. They didn't used to collect biospecimens, but now they've added to their kind of registry and study cohort a biospecimen because they have tremendous data about patient outcomes and exposures and are now able to integrate genetic risks into that. Sometimes, yeah, and so that's an example of a clinical trial. And then many times, it can be just a useful tool to complement other kinds of bioinformatic projects that are going on. And so sorry, this is so small, but this is our triage in the NICU, which is basically that if we approach everyone for participation in research, and if there's kind of no concern for an underlying genetic diagnosis, they're in with RSV bronchiolitis. I don't think it was their genes that gave them the virus. It was probably their lovely but slobbery 2-year-old sibling, right? We would approach them for the biobank participation where we would take excess samples or whatever they would like to consent to. If someone came in with a particular difference on their brain MRI that makes us very likely to think that it's a genetic diagnosis, but let's say it was 5 years ago and we only did exome sequencing, we then would have consented them for research genome sequencing, right? But if they had a very specific condition where perhaps their genome sequencing now was negative, but they have a muscular dystrophy, and we think that RNA sequencing is actually going to be what shows us where the pathogenic variant is, then we actually have them enroll in the Morton Center where there's very prospective and purposeful testing of things like blood RNA or doing long read sequencing to look for those missing variants. And so that's where we try to have everyone have the maximum benefit from their participation. So if it's a diagnosis that the Morton Center is going to go after, we have them consent there, because I think there's potential participant benefit. But if Morton Center declines, then we do our genetic testing on a research basis through our project. So we take any baby, we take anything they want to give us. And then this was just our example project, and this is my last slide, is just that there was a fellow who was really interested in the immune response to viruses. And we're one of the only NICUs in the state that will take babies who've been home and babies who have known infectious diseases, right? NICUs are usually only if you were born in that hospital do you get to enter. And so we're a unique place to get to see babies who have severe impact of respiratory illnesses. And so the fellow was able to use our existing IRB. We've been able to collect the swabs that she would like and store them in the lab. And that probably saved her a year of regulatory applications. And so that was nice. So I think biobanks are an important piece of our biomedical research. There are some exemplar ones that have really transparent policies. If you're ever thinking of starting one or just want to know more about how they tick, they have lovely documentation. And sometimes small local biobanks might still be useful for the particular question that you're looking in, and whether this is a community foundation that has made their own registry and now has biobank samples, or whether it's at a hospital, they could be nice resources. So thanks so much.
Thank you so much. If we have some quick questions, just walk up to the microphone.
Thank you. Thank you for the talk. Given that these biobanks are meant to last forever, how do you kind of achieve the financial stability or health of the biobanks?
Yeah, I think the big ones do it as we saw, right? UK Biobank used to be application to access, and now there's fee for access, right? And so I think a lot of how people are becoming sustainable is using fee for access to keep those lovely automated machines going. I think a lot of biobanks are also partnering with groups who pay for access to the data on large scales, right? So if you're someone who has a commercial interest in a particular gene, being able to have access to do the proteomics to go along with all the diagnosis. I think that we've seen partnerships like that, right? Like Regeneron has many ways in which they're able to commoditize the data from their sequencing cohort and biospecimens to be able to make it sustainable. But fundamentally, there's no, you could imagine any of them collapsing, and then it would just be up to the community to find someone who would be able to kind of take over the specimens that have value. I think much of the data, right, because there's the data infrastructure and there's the biological specimens infrastructure. And in some ways, those are able to be separated too. But it does seem that in our current climate, that there might be some changes, either in requiring access to do many of the experiments or paid access to do many of the experiments that we used to do for low or no cost, or some of them not being sustainable. Like even at our hospital, right, we downsized all of our samples pretty significantly to be able to keep just the highest yield ones.
Great. Thank you so much. That was wonderful. And I think we'll stop there for time. Thanks, everyone. Thanks, Sarah.